Hello, Assalamu alaikum. So students, today we are going to discuss the process of the glycolysis. Glycolysis, this is the process which occurs in the cytoplasm, and it is common both in aerobic and anaerobic respiration. Aerobic respiration is the process which occurs in the presence of the oxygen, whereas anaerobic respiration occurs in the absence of the oxygen. So glycolysis is common in both of these two types of respirations. Glycolysis completes in two steps. The first step is known as the preparatory phase, and the second step is known as the oxidative phase. Glycolysis is the process in which the glucose, a 6-carbon compound, is broken down or is converted into two molecules of a 3-carbon compound, which is known as pyruvic acid. This is a 3-carbon compound. So let's start with the process. What happens in the glycolysis is that the glucose, which is a 6-carbon compound, utilizes one ATP molecule that is converted into ADP and inorganic phosphate, and the glucose is converted into its isomer, which is called as the glucose 6-phosphate. Then the glucose 6-phosphate converts into its isomer, which is known as fructose 6-phosphate. The fructose 6-phosphate again utilizes one ATP molecule and converts into ADP and inorganic phosphate and changes into its another isomer, which is known as fructose 1,6-biphosphate. As it is a 6-carbon compound, so in the presence of another enzyme, it changes into dihydroxyacetone phosphate, which is a 3-carbon compound, and phosphoglyceraldehyde, which is also a 3-carbon compound. Both of them are interconvertible into each other. This is the first step of glycolysis, in which the glucose forms the substrate for the formation of the pyruvic acid, which is known as the PGAL. Now, as I said that the DAP and PGAL are both interconvertible into each other. So what happens that the DAP converts into phosphoglyceraldehyde, and now we have two molecules of phosphoglyceraldehyde. Now the second phase, which is known as the oxidative phase, will be started, and the PGAL now combines or reacts with the two inorganic phosphates. The two NAD molecules will be oxidized into two NADH2 molecules, and it is converted into 1,3-biphosphoglyceric acid. It is also a 3-carbon compound and two molecules are formed. Note, I'm writing the two molecules, that's why, because we have two substrates of the PGAL. Then the 1,3-bi-PGA molecule releases energy. Two ADP molecules reacts with two inorganic phosphates, and two ATP molecules are generated, and it is converted into 3-phosphoglyceric acid in the presence of the specific enzyme. This 3-phosphoglyceric acid undergo hydrolysis. One water molecule is released, and the 3-PGA is converted into phosphoenol pyruvic acid. The phosphoenol pyruvic acid then again releases energy. Two ADP molecules react with two inorganic phosphates, and two ATPs are generated again, and the phosphoenol pyruvic acid is converted into pyruvate or pyruvic acid, which is a 3-carbon compound. So now with this, the oxidative phase of the glycolysis is completed. At the end, we have 4 ATPs produced. But 2 ATPs are utilized in the preparatory phase of the glycolysis. So we'll minus 4 minus 2, and then the net gain of ATPs in glycolysis is 2 ATPs. This is the whole process of the glycolysis. So giving you a quick summary, that glucose utilizes ATP molecule and converts into glucose 6-phosphate. Then the glucose 6-phosphate converts into fructose 6-phosphate. Fructose 6-phosphate then again uses an ATP and changes into fructose 1,6-biphosphate. Fructose 1,6-biphosphate, as it is a 6-carbon compound, it splits into 2 3-carbon compounds, the dihydroxyacetone phosphate and phosphoglyceraldehyde. Both of these are interconvertible into each other, and DAP is converted into PGAL. So now we have 2 substrates of the PGAL. The PGAL react with 2 inorganic phosphates. 2-NAD are converted to 2-NADH2, and it is converted into 1,3-bi-PGA. This 1,3-bi-PGA releases energy, and ATPs are converted into 2 ATPs. And it is the 1,3-bi-PGA is converted into 3-PGA. Then this phosphoglyceric acid undergo hydrolysis, and it is oxidized into phosphoenol pyruvic acid. The phosphoenol pyruvic acid then generates 2 molecules of the pyruvate, which are a 3-carbon compounds. Then we have the net gain of ATPs is 2 ATPs in glycolysis. If you like the video, then thumbs up and like the video, share the video, and please subscribe the channel. Thank you.